I thought it would be a great fun to take advantage of this opportunity and talk about the International Year of Crystallography and actually kick off our celebratory events. The next presentation in this vein is going to be given by Eddie Arnold. He'll also give a presentation that will help us mark the International Year of Crystallography 2014, 100 years after the first X-ray structure was produced by the Braggs, as you'll see. I'll try and alternate between the two sides of the room. This is another experiment. We've tried both orientations. There was a push to return to this east-west orientation. Let me know if it works, and if it doesn't, we can always go back. First, a disclaimer. This is informed by my journey. I want to tell a story that involves the path that a recovering physicist has taken over the last nearly 40 years that involves structural biology, medical care, entrepreneurial activities. What I want to do is to explain how I think a very glacial and largely peripheral scientific art form transformed itself into the mainstream tool of modern biomedical research that it is. And I'll set that in context. So in 1980, when I graduated from the University of Western Ontario in London, Canada, where my parents moved when I was about 16 years old, physics student, crystallography was incredibly slow. And I'll tell you exactly how slow it is a bit later on. I went, having been bitten by the biology bug, went and did a DPhil in what we would now call structural biology at Oxford, as Helen said. Realized as a physics student I didn't know any biology, and I was not going to be useful if I didn't remedy that deficit. So I went to Harvard Medical School, did the MD. I didn't want to sleep, so I did postdoctoral work with Greg Petzko, and then I had a further period of 4 years with no sleep, where I did internship and residency in internal medicine at the Brigham and Women's Hospital in Boston. And I was also a postdoc with the Nobel laureate William L. in Lipscomb, and I'll show you the structure that I determined in the Lipscomb lab. And then I had the great fortune to be able to go down to Rockefeller University in New York City, where I ultimately served as the Richard and Isabel Furlow Professor. I was a member of the Howard Hughes Medical Institute and had a great run studying the structural biology of gene expression. And then I underwent a transition to the dark side. Got involved in industry, went to be CSO at SGX, took the company public, sold it to Lilly after the stock price tanked when we had a bad clinical outcome that I talked about a year ago in this room, and had a very interesting 3.5 years at Lilly, integrating the SGX drug discovery platform into Lilly drug discovery. And then they invited me to move to Indianapolis, and I politely declined. And I attempted retirement. That failed, as Helen knew that it would. And she and Ken Breslauer were right there as I was failing retirement and said, but look at the opportunity here at Rutgers. New building, protein data bank, integration of 2 medical schools, the School of Public Health, Cabham, Waxman, etc. Why don't you come to Rutgers? So, long story short, I'm here and having a great time. So the story begins back in 1913 with the publication of the structure of the diamond by a father and son team of pioneering physicists, the Braggs, senior and junior. They get the credit for the establishment of this law: n lambda equals 2d sine theta, which is the diffraction condition for an X-ray bouncing off successive rows of atoms or proteins or molecules arranged in a lattice. And this was what X-ray crystallography looked like experimentally in 1912. That was an X-ray spectrometer. Hard to imagine that versus the synchrotrons of today. So a word about protein crystallography and an attempt to demystify. So everyone in the room, I think, I'm confident, is familiar with a light microscope. So we have a specimen. On a sample stage, visible light passes through it. It's diffracted. It's collected by a pair of lenses and ultimately magnified, and a virtual image of the enlarged image of the amoeba, in this case, is presented to the retina, and we're able to make enormous advances, of course, as a consequence. This is actually an analog computer. It involves the calculation of 2 successive Fourier transforms, and the importance of a Fourier transform of an object is that you get back the object. So the function is the inverse of itself. That, coupled with a magnification step, allows one to do imaging. So X-ray imaging, X-ray crystallographic imaging, is done with calculation of the Fourier transform. One half of the experiment, the first Fourier transform, is calculated by in analog fashion by a machine, an X-ray diffractometer, and what we record are the intensities of the Fourier transform, which is a reflection of the distribution of electrons in the crystal, in the crystal lattice. Unfortunately, when you do this experiment and you record the intensities of the Fourier transform, you throw away 50% of the information, because you recall that light, X-rays, light, of course, has a property of both amplitude. It's a wave, and has the properties of both amplitude and phase. And the phase information is sacrificed at this stage. Now, there are tools that allow one to estimate the phase angles for each of the reflections, the recorded spray of X-rays coming from the crystal. And with these measurements and those estimates, one can compute in digital fashion now, as opposed to in analog fashion. One can compute the second Fourier transform and reproduce a magnified image of the 3-dimensional distribution of electrons in the crystal, in the protein, in the small molecule, and then the human eye can interpret those the peaks in that distribution in terms of an atomic model. And remember the word model. This becomes important. So this was just after the turn of the 20th century, 1913. Bragg and Son use a very crude incarnation of this technology to determine the 3D structure of the diamond, and they go on very rapidly to determine a whole series of other structures of small crystalline solids. The breakthrough for us as biologists interested in a structural view and interested in the relationship between structure and function comes first from the work of Northrup, who purified pepsin to homogeneity using serial crystallization. And he showed that this enzyme pepsin was a protein by analyzing the contents of the crystal. About a decade later, he was able to isolate and crystallize the first virus, a bacteriophage, and he documented that it was made both of protein and nucleic acid. So here's the first clue that the genetic material was going to be, the genetic information was going to be stored in nucleic acid. And for that, not surprisingly, he got the Nobel Prize. My connection to Northrup is that he was a longtime Rockefeller faculty member, where he enjoyed, particularly before the Second World War, working in an institution where you were not allowed to apply for a grant. Freedom. You got money from the central administration to do research. You were free to do whatever you wanted. And so people like Northrup and others, of course, did great things over the years. Capitalizing on Northrup's studies of pepsin, Dorothy Hodgkin, working with one of the giants, the early giants in crystallography, J.D. Bernal, noted Marxist amongst other things, recorded the first X-ray diffraction pattern of pepsin and showed that it was ordered, that there was clear evidence of 3-dimensional order and hence a well-defined structure. She went on to make very substantial contributions in small molecule crystallography with the determination of the 3D structure of benzyl penicillin in the 1940s following the industrial preparation or industrial manufacture of penicillin. And for that, among other things, she received the Nobel Prize. And structures of this type are present in the Cambridge Crystallographic Database, which has its U.S. operations here on the first floor, led by Paul Davey, who's in the audience with his team of specialists in small molecule crystallography, including Sharm, I don't remember your first name, sorry, who's the first of the U.S. operation. In the background, all this time that she was working on benzylpenicillin, vitamin B12, etc., Dorothy was working on the structure of insulin, the protein hormone that governs blood sugar. Interestingly, insulin was discovered by Sir Frederick Banting, the name of the secondary school I went to, the high school I went to in London, Canada. Insulin was commercialized by Eli Lilly and Company in the 1920s, immediately following the discovery in collaboration with Banting and Best. Here's a portrait of Dorothy, her arms a blur, doing 20 different things at once. And I think many of the faculty can sympathize with her, perhaps. In her office in the Department of Chemical Crystallography at Oxford, with an atomic model of insulin in the foreground. I met her as a first-year graduate student at Oxford, a towering figure in protein crystallography, and also a great source of inspiration and encouragement for students. Now, I mentioned the phase problem and the challenges that presents to us as protein crystallographers. In 1949, Byfet, a crystallographer working in the Netherlands, documented that if you have atoms in crystals that absorb X-ray energy, this breaks the symmetry of the diffraction pattern, and that broken symmetry actually allows one to overcome the phase problem just by measuring the intensity. So even though you've thrown the phases away, one can actually recover the phases, the absorption effect in crystals. This was first demonstrated conclusively for proteins by Wayne Hendrickson and co-workers, with particularly Martha Teeter working on crambin. And here is a rendition of one of the disulfide bridges in crambin from Irving Geist. I'll say more about him in a moment. It was the anomalous scattering, the energy absorption of these sulfur atoms in the crambin crystal that allowed the phase problems to be overcome simply with intensity measurements. This didn't happen until about 1980, and there was a great deal of activity in protein crystallography beforehand, the reason being that another tool was developed by Perutz and others in Cambridge called multiple isomorphous replacement that involved making a defined chemical change to a protein, adding a heavy atom such as platinum or mercury. And with this, there was another way of overcoming the phase problem, but it involved measurement from multiple crystals and is fraught with a great deal of difficulty. Today, we can routinely incorporate selenium into proteins through the use of selenomethionine, and most of the new crystal structures that involve de novo phasing today are the result of this incorporation of selenium and the use of synchrotron radiation. The very first structure of proteins that emerged in the late 50s, early 1960s was that of myoglobin, the oxygen storage protein found in our muscles. This is what makes meat red. It's the heme group that binds the molecular oxygen that makes in the myoglobin that makes our muscles red. This is not a very pretty structure at first glance. At low resolution, it actually looks like a pile of dog turd, as you'll see in a moment. It's entirely alpha helical. That was a huge plus, because even at low resolution, even with a modest resolving power of the X-ray method, one can get some sense of what was going on. Ultimately, of course, we had this all-atom structure of myoglobin that was painted by Irving Geist, and these illustrations appeared in an article in Scientific American published in 1961 that has influenced a whole generation of protein crystallographers. Many people have said to me, I read this article in my parents' old Scientific Americans, and that's what convinced me to do crystallography. No molecular graphics in those days, of course. Shortly thereafter, Perutz, the inventor of the MIR, multiple isomorphous replacement method, determines the structure of hemoglobin, the oxygen carriage protein found in our red blood cells. This was the first evidence of oligomeric structure in proteins. So 2 subunits of alpha, 2 subunits of beta to form a heterotetramer that exists in 2 dominant conformations, oxy and deoxy. And what was interesting about it was it looks like 4 myoglobins put together in a symmetric arrangement. So there's already evidence of nature using the same protein shape to do two different but related jobs, and I'll come back to that in a moment. So I want to say something about use of language briefly. I will talk about structure and I will talk about the structure, but what I really mean when I say structure is an atomic level structural model derived from an interpretation of experimental data. And when crystallographers who over the years have produced 90% of the contents of the protein data bank, when they always talk about the structure, the atomic level structure. And what they mean is that for every atom in the structure, there's an X, a Y, a Z coordinate, a B factor, which describes how much the atom appears to be moving around in the crystal, and the occupancy, if it's not present at 100% occupancy, as might be the case for a ligand. But what they really mean is the average X, Y, Z, B factor occupancy for each atom determined over the volume of the crystal and for the duration of the X-ray experiment. So it's a blur. There is no, what they're actually measuring is a slightly blurry electron density map. They're interpreting it in terms of atomic positions. And it's probably the case that the X-ray structures that are in the PDB never actually exist in nature because it's an interpretation of the average electron density over the volume of the crystal, the course of the experiment. It doesn't mean that it's not useful, but the precision of the atomic positions at best is about 1/10 of the resolution limit, so about 0.2 angstroms typically for the XYZs. Because protein crystallography, unlike small molecule crystallography, is actually a slightly underdetermined problem in terms of the ratio of measurements to the number of parameters that are used in the refinement process that arrives at these X, Y, Z, B occupancy, etc. So there are these caveats. These structures are really useful, but they are not the same as the small molecule structures where you know bond lengths to within 1/100th of a degree, you know, bond angles to within 1/100th of an angstrom, bond angles to within 1/100th of a degree in the best structures. So this is what myoglobin looked like initially. It really did look like a pile of dog turd, right? And there was a plate in the middle of it. That plate was the heme group. So the only way we had in the 1960s, 1970s of looking at proteins was Irving Geis. He came to the lab where a new structure had been determined, and he painted a portrait. He got a commission typically. He got one from me. He painted a portrait, and then these became the tools that we used for actually looking at structures. And there's no doubt that these were inspiring portraits. But today we have absolutely amazing molecular graphics that came via flight simulators to the gaming industry and allow us to draw things like this routinely. So here's a model of the chromatin fiber with the DNA snaking around the nucleosome core particles here in this model of the fiber. So we've gone from Geis to military applications that spilled over into the civilian regime to the gaming industry, which is now revolutionizing computing and revolutionizing graphics. Geis also painted the structure of the first enzyme, 1966, that of lysozyme, the effort led by my PhD supervisor, the late Sir David Phillips. This is a single domain enzyme that's responsible for catalyzing hydrolysis of 1,4-beta linkages in peptidoglycans. It includes both alpha and beta structure, both of which were predicted to exist before the very first crystal structures emerged. And shortly after this structure was published, there was a few enzyme structures came, one from Yale, one from Harvard, that I don't have time to talk about. Also coming from the Oxford lab subsequently was triose phosphate isomerase. And it's got a different shape from myoglobin, hemoglobin, from lysozyme. And it was the case that when structures of macromolecules emerged in the 1960s, 1970s, everyone was fascinated to know what they looked like because we had seen so few. And what we began to appreciate was that there was diversity of domain structures. But as things progressed, we quickly learned, well, perhaps not so quickly learned, but ultimately began to appreciate in spades that these shapes are being used over and over again by nature. And so when we ask the question today, what does it look like, we mean something totally different. We're not expecting that it's going to look like something new. We're expecting it's going to look like something we already understand, we've already seen, but as a result of gene duplication, divergence, the acquisition of a new biochemical or biological function, you're using the same machine to do a slightly different job. And this is particularly true of the members of the triose phosphate isomerase family, one of the most abundant folds, globular protein folds in nature. So you can see the tin barrel here viewed side on, and you can see there's another domain capping it. This is an enolase, a structure we determined as part of my involvement in the NIH-funded Protein Structure Initiative. So this is where the catalysis takes place, but the selectivity for the substrate is determined by this capping domain. There are literally tens of thousands, if not hundreds of thousands, of enzymes in nature that look like this. Tin barrel on the bottom, but a variation on the domain theme determining the specificity of the enzyme. So when we see a new structure like this, we're seeing a familiar friend. Sometimes sometimes we see a familiar friend here, sometimes we see something completely new. The important thing is that what we're able to do is to capitalize on the wealth of information that is available in the protein data bank to better understand both the relationship between protein structure and function, and also, of course, learn a great deal about evolution. So the origins of the PDB were humble. 7 structures in 1971, a consequence of a Cold Spring Harbor meeting at which the community, very interesting, decided that data sharing in protein crystallography was essential, and that they would catalyze the creation of the first open access digital resource in all of biology. This is before anybody knew what open access meant. They were just sharing we were going to share the information. It's an archival data bank. In 1999, as Helen told you, she did not listen to the advice I gave, and I have no memory of giving that bad advice, of course. I only remember the good advice that I gave. 1999, with the original home of the Protein Data Bank in the United States drowning under what was at that time viewed as a deluge of structures. It would just be a couple of months in terms of today's science on an annual basis. The NSF, NIH, DOE made the very shrewd decision to move the Protein Data Bank to Rutgers, put it in the care of the Research Collaboratory for Structural Bioinformatics, and the census today in the data bank is approaching 100,000 entries. 90% of them are X-ray crystallographic. In 2003, Helen led a critical effort to avoid the balkanization of the archive by getting data centers in Europe and Japan and the US to agree to form the Worldwide Protein Data Bank, which maintains a single archive of all macromolecular structures that have an experimental basis. Here's the logo of the Worldwide Protein Data Bank and its URL. This was a key move, and it now sets us up for the opportunity that in a few years, as the pace of science in China and in India develops, there'll be a PDB China, a PDB India. One could even imagine with the emerging economy in Brazil, synchrotron radiation being established there, and the objectives the Brazilian government has to establish a drug industry. So there could even be a protein data bank Brazil. Recent innovations among the three existing data centers are creating a global system of deposition so that it will be possible to have franchises, just as McDonald's does, a standard way of doing things. So this was my first deposition of a completely new structure to the Protein Data Bank back in 1989. This is leucine, or 1990 perhaps, leucine aminopeptidase. It's a hexameric structure with 3-2 symmetry that is responsible for cutting off the N-terminal first amino acid in an extended polypeptide chain. And the way it does that is that rather than indiscriminately cutting off the N-termini of globular proteins. The active sites are actually in the middle of this hexameric cage. So only an extended polypeptide can get in, and the first amino acid can be chewed off. And of course, it's got a preference for the hydrophobics. This structure was determined from 10,000 cow's eyes from a natural source. And I swore at the end of this experience I would never work on a project again that involved a protein that was not recombinant. But this was 1989. The recombinant production of proteins was in its infancy at that stage. One way of overcoming it, of course, was actually total chemical synthesis of proteins. This is the structure of the HIV protease. That was determined in a collaboration by Vlodauer and Kent. Kent, actually a scientific son of Bruce Merrifield, solid-phase peptide synthesis at Rockefeller, actually synthesized this dimeric enzyme. Structure was determined by Alex Vlodauer, put into the public domain. This sets the stage for the discovery using structure-based drug discovery of protease inhibitors that are now at the heart of, if you'll excuse the pun, HART, which is highly active retroviral therapy. It's these cocktails that are keeping patients with HIV/AIDS alive today for very long periods. Another critical thing that happened for the field was the emergence of genome sequencing. So in '95, you recall, some of you will recall, that some of you weren't born then, that Haemophilus influenza genome was published with a map that looked like this. This created enormous opportunities for us structural biologists, targets galore. Absolutely amazing. The number of proteins that we were going to be able to get access to as a result of genome sequencing. In the early 90s, recombinant production of proteins began to be routine. A less understood contribution of the genomic revolution was liquid handling robots. This work was done by liquid handlers. These liquid handlers are now used routinely in crystallography labs to do with much higher precision and a much lower error rate the tasks that were historically done by individuals. So here's a crystallization robot from SGX Pharmaceuticals, an old-fashioned one by today's standards to be sure, but a great breakthrough at the time. I mentioned leucine aminopeptidase and how difficult it was to work on something that was coming from a natural source. In the old days, and I was on that very tail end of the old days, what was done by crystallographers was to purify crystallizable proteins from either bacteria or tissues. Today, what we do is we decide what we want to study, and we render the protein crystallizable by making them look like something like ribonuclease or lysozyme or carboxypeptidase A, the very first enzymes that were determined, which are very, very globular, no floppy bits. So here's a rendition of a protein, sort of domain rendition of a protein kinase polypeptide chain, one we worked on at SGX Pharmaceuticals. So it's got what's predicted to be random coil, a globular domain here, non-catalytic, more random coils, the catalytic domain that's responsible for the kinase activity, and another piece of random coil. And Gianmarco Trigiano, who worked with me at Rockefeller, could tell you immediately that this thing full length will never crystallize, as was the case with his EIF4E. The floppy bits get in the way. Proteins that look like this don't crystallize. Proteins that look like this crystallize. So the key is to decide what you want to study, what the problem is, and to try to engineer the protein in such a way that you can get something that's going to crystallize. A key determinant of crystallizability, of course, is solubility. So by cloning out smaller segments of the DNA encoding this protein using distinct forward and reverse PCR primers, one can generate a whole different series of N and C terminal constructs. It's also possible to do internal deletion constructs, and these can be expressed in E. coli and tested for solubility. And not surprising in this instructive example, the PCR primers that respect the N and C terminal boundaries of the globular catalytic kinase domain yield a protein in E. coli that's highly soluble, one that you can frequently go on to crystallize and to determine the 3D structure of. So this protein engineering involves going back to the past in a sense, because we're trying to make something that looks like ribonuclease. It is reductionist, and that is an issue. but one does what one can, and then there are other tools like cryo-electron microscopy, in which we're doing a search jointly with cell biology and neuroscience, where one can look at much larger assemblies that you might not be able to crystallize. And of course, there's also NMR that will allow you to look at things that don't crystallize. So I mentioned the critical role that physicists played in the birth of protein crystallography, the whole science of crystallography, and this type of science, the discipline of physics, continues to contribute at a very high level. So starting in the 70s, but really coming of age in the 80s and 90s, cryogenic crystallography, reducing radiation damage first on the home source and ultimately at synchrotrons, involving the delivery of refrigerated nitrogen gas that is bathing the crystal that's maintained here at the head of this pin at a temperature on the order of 100 Kelvin, greatly reducing radiation damage. Crystallographers have also benefited from the emergence of facilities for synchrotron radiation. So this is the Advanced Photon Source just outside Chicago at Argonne National Lab, where SGX Pharmaceuticals, now Lilly, had their own private beamline right about there. These are intense, bright, and importantly, tunable X-ray sources that allow us to take advantage of the contribution that BiFIT made back in 1949, breaking the symmetry of the diffraction pattern, overcoming the phase problem, and delivering data at a rate which is simply unparalleled. Using the Lilly beamline now, one can at the APS, one can now get data from a protein crystal and determine a structure in less than 5 minutes. That's routine. The data collection, less than 5 minutes. That's because of the emergence of detectors of this type. This is this is essentially direct pixel array detection of scattered X-rays. This little beast costs about $2 million, comes from a company in Switzerland called Dectris. And it has revolutionized, is in the process of revolutionizing data collection at synchrotrons. This is one being installed on a beamline at the Advanced Photon Source that is jointly operated by academic institutions here in the Northeast. Let me return to the PDB and make some comments about the importance of data sharing. One of the great things that came from even the very early efforts at data sharing that the PDB undertook was an understanding of what proteins really looked like. So this, you can trace this all the way back to G.N. Ramachandran and the emergence of the Ramachandran plot at the Indian Institute of Science in Bangalore. So that's been followed up by a whole series of tools which are now used routinely to assess the quality of protein structures. And we're really asking, does this new structural model look like the other structural models that are in the protein data bank? Where are the similarities? Where are the differences? And how does the quality of the structure compare? And just in the last couple of days, I think the worldwide protein data bank has gone online with the delivery of very detailed validation reports on every structure in every structural model in the PDB that allow one to assess at a glance what the quality of the structure is. If you're on the blue end, you're above average. If you're on the red end, you're below average. So the Burley lab structures were all above average, like the children of Lake Wobegon. So not only do we get some sort of global sense of how good the protein structural model is, but we also get a sense if there are issues at particular places. So there are plots by residue, and where you see red dots here, there's a suggestion that there's a discrepancy between the experimental electron density and the atomic model. So as a biologist, you can view this in two ways. I don't want to go there, it's not well defined, or, wow, the crystallographers don't know what's happening here. Here's an opportunity to make a mutation and study the important role of that particular amino acid if it's sitting in an active site or something like that. So lack of resolution in X-ray crystallography is not always a bad thing. With every new field, there are teething pains. And a series of honest mistake, wrong structures appeared in the crystallographic literature through the 70s and 80s, much less so now. And they gave rise to some very important tools that we're now using routinely to assess protein structure quality. And they include David Eisenberg's threading, the emergence of the free R factor, and very prudent use of the tool of density modification, which helps. It's one of the computational tools that we use to take advantage of Bifet's early observations. And this has culminated in this enhanced WWPDB validation of the entire system. So now I want to take you through a whirlwind tour of some of the really cool structures that have appeared over the years. Some of them are in the nucleic acids database, the NDB, some of them are in the PDB. So here is Helen's first structure. It's UPA, and it was designed to produce Watson-Crick hydrogen bonding. That was the theory. It actually did something very different, as you can see, but nonetheless very interesting, and set the stage for a whole series of important crystallographic contributions to our understanding of nucleic acids. The next landmark I want to refer to was the structure of GPC, which showed the first evidence by X-ray crystallography of what we know as Watson-Crick base pairing, as you can see in the images. Watson-Crick base pairs abound in this structure of yeast phenylalanine tRNA, which was published in the 70s, both by labs in Cambridge, Mass. And Cambridge, England. And this foray into the translation machinery set the stage for the amazing work that's gone on using both X-ray crystallography and cryo-electron microscopy to study the central machine in the translation system, the ribosome. So we had first structures of ribosomal proteins coming from people like Venki Ramakrishnan, then ribosomal subunits, and now whole ribosomes. And just a few years ago, Venki with Tom Stites and Ada Yonath, who pioneered work on crystallography on ribosomal subunits, shared the Nobel Prize in Chemistry for essentially this structure and many others that look like it. And we know an enormous amount about this machine today as a consequence of the advances that have occurred, beginning with, in terms of RNA crystallography, beginning with the work that Helen did as a graduate student at Pittsburgh on UPA more years ago than she probably cares to remember. First crystalline structure of an interesting-looking DNA molecule came from Dick Dickerson out of on the West Coast at Caltech at the time, I think he was. The Dickerson dodecamer, so 12 base pairs. So this showed bDNA. This was the first formal crystallographic proof that Watson and Crick were right. It's a beautiful structure showing base pairing, showing tilting of bases, sliding, rolling, all sorts of interesting things which we've now come to know and love. And we saw with particular fascination in this structure, which was the one I got tenure for at Rockefeller. It's the Tata box binding protein interacting with the Tata element, and it puts two kinks in the DNA, one here, and puts the DNA through a 90-degree bend. And it's this platform on which the RNA polymerase II machinery is assembled for Pol2 transcription from Tata-containing promoters in organisms from yeast all the way up to man. And I should say that this protein is very highly conserved, so the sequence identity from human to yeast is 80%. The sequence identity from archaea to human is 50%. This is a protein that wasn't allowed to evolve, basically, because doing so would threaten the livelihood of the cell bearing such a mutation. We've gone from this very simple, but at the time very, very challenging structure to structures like this, determined in Roger Kornberg's lab of yeast RNA polymerase II, and you see a rendition here of the structure from David Goodsell's work showing both the DNA template and the emerging pre-mRNA. So we've gone from relatively simple transcription factor DNA complexes 20 years ago to structures of this type, and with the advances that are occurring in cryo-EM, some of you may have seen some talks over the last week or so showing how transcription factors can be incubated with the polymerase and nucleic acid to build up large mechanistic mechanistically active complexes of the transcription machinery that can be studied by cryo-electron microscopy. Another great advance that came courtesy of biochemists and structural biologists was structures of integral membrane proteins. So we start with a protein from the natural source, the reaction center that came out of the Huber lab in Martinsried, just outside of Munich. And that was followed up many years later by a gentleman I recruited to Rockefeller who went on to win the Nobel Prize. This is Rod McKinnon's structure of the potassium channel, a very, very simple two-helix tetrameric arrangement that explained potassium ion selectivity of this channel, and this particular portion of the structure of this potassium channel is replicated in all of the potassium channels that we're able to identify in nature. It is the selectivity filter that ensures that only potassium goes through the pore and shows just how good nature is at reusing shapes that work. A terrific advance came last decade from work at Stanford, work at Scripps, first seen with the beta-adrenergic receptor structure. This is the first structure of a recombinant GPCR. It has a beta blocker bound to it. Some of you are probably taking beta blockers to control blood pressure, heart rate, etc. And in fact, approximately half of all the drugs that are approved by the FDA are directed at GPCRs, proteins with this fold. I was very proud of the fact that just as I was leaving Lilly, a team that I had persuaded the organization to establish that was dedicated to studies of GPCR structure produced the structure of a novel human GPCR at about 2 angstrom resolution with a Lilly compound bound in the binding site, setting the stage for structure-guided drug discovery of GPCRs at Lilly. Upper management was very receptive to my proposal. My colleagues, my SGX colleagues, former SGX colleagues were just profoundly unhappy and kept saying, but what if we fail? What if we, after 3 years, what if your prediction doesn't come through? Half a dozen people might be fired because you put them in a lab and told them do nothing but work on this system and get it working for Lilly. What happens when Lilly loses patients? Well, Lilly didn't lose patients because they knew how important it was. And just over 3 years after I made the pitch to upper management in drug discovery at Lilly, they produced this structure, and suddenly everybody's all smiles. And all the people who criticized me roundly for forcing them to take the risk suddenly remembered that it was their idea. So remember this. Success has 1,000 parents. Failure will always be an option. Always be an orphan. Always. Apologies for the delivery. So the other great thing that's come along, and it's stunning actually when you think about it in protein crystallography, is that one can crystallize viruses. So here's the structure of the first virus to be determined by X-ray crystallography. Work of Stephen Harrison on tomato bushy stunt virus, beautifully rendered here by Kathy Lawson, who's the local PDB expert on virus structure. This set the stage for a whole series of structures of other plant viruses, structures and then structures of animal virus, such as the work that David Stewart and colleagues did on foot and mouth disease virus. This is not something we see in the United States, but it is a significant economic problem in the UK and in Europe. Excuse me. And they are using knowledge of this virus to actually create a vaccine that they hope to use with cattle to prevent this potentially devastating pest from threatening the livelihoods of farmers. So that was amazing in its own right, a huge, huge structure. But what came last year, not as a result of X-ray crystallography, but building on the results of X-ray crystallography from Angela Gronenborn's lab in Pittsburgh, was the structure of the HIV capsid. So this is the nucleocapsid of the virus that causes the human disease AIDS, and they determined this 3D structural model from an integrative approach. They had X-ray structures of the hexamer. They had an X-ray structure of the hexamer here. They used cryo-electron tomography to establish the arrangement of all of these hexameric and pentameric segments of the surface of the capsid, and then working with, is it Klaus Schulten? Yeah, working with Klaus Schulten, thank you. They used a molecular dynamics approach to sort of move these things all around so they stopped banging into one another and produced a model that made sense, that consisted of more than 2 million atoms. This transiently broke the PDB software, but where's Zhu Kang? Zukang, in a week, recoded there was basically an array limit. No one had ever thought there'd be a structure of more than 2 million atoms, right? How's that ever going to happen? Well, there is. And in short order, the software was extended so that we can now handle structures of any size. And that sets the stage for going beyond this remarkable discovery of the determination of the HIV capsid structure. So a word about productivity. I'll try and finish fairly quickly. So in 1980, when I joined the laboratory, the prosaically named Laboratory of Molecular Biophysics at Oxford University, it was taking a team of 4 or 5 people, 4 or 5 years, i.e. 20 person years, to determine a protein structure after there was an abundant supply of crystals. Shocking. Today, it takes less than 30 person days to go from an idea to a completed high-res structure, and the evidence I'll cite for that was the SGX structure of the SARS protease, which we published in the New York Times, and deposited into the protein data bank. Of course, this was at the height of the SARS crisis, and we thought we could do 2 things. We thought we could do a public service by getting the structure of the SARS protease, a potential drug discovery target for SARS, and we, of course, also thought there might be some economic benefit to us down the line. We got kudos, but we didn't get any dollars. But it showed that a well-organized team can go from an idea to a structure of a protein, a good-sized globular protein, in the space of less than a month. And this has been reflected across academia, across the industry. We're now getting in excess of 850 new depositions to the protein data bank. They're coming from these sites. All of the inhabited continents, nothing from Antarctica yet, unfortunately. The users, so about 1 million downloads a day of atomic coordinates, are again coming from every populated continent in the world. And you can see the growth in depositions, both actual and projected. And we anticipate that there's going to be essentially a quasi-linear increase in depositions to the protein data bank on an annual basis. Between 2013 and 2008, there was a 50% increase nearly in the number of depositions on an annual basis. And no reason to believe this is going to do anything other than go down. Anything other than keep going up. And of course, the complexity of the structures is going up all the time, and we're also getting more and more structures from cryo-electron microscopy, which is extremely exciting because we're seeing structural models for things we couldn't crystallize. I estimate that industry has another 100,000 structures in its own archives, spread among all the companies. SGX Pharmaceuticals was part of a move to pioneer fragment-based drug discovery, guided by structure. And the very first fragment-based drug has been approved for melanoma. It was taken from Plexicon by Roche and has been approved for melanoma. This is the structure of a MET inhibitor that we put into the clinic that crashed and burned in phase 1, but an analog of it is now safely in the Lilly drug candidate pipeline. All major pharmaceutical companies are using fragment-based structural approaches for drug discovery. And one of the things I hope to do moving forward with the PDB is to try and extract some of these structures of protein ligand complexes from the industrial databases, because there's a great deal of data there that will be of benefit to academics trying to understand the energetics of protein ligand complexes, etc., trying to understand how to do better drug discovery. The future, I'm going to say 2 things about the future when I close. The first is moving towards what we call integrated structural biology, hybrid structural methods, and I'm specifically singling out the challenge and the opportunity created by the nuclear pore complex. This is a yeast nucleus, electron micrograph, colored accordingly to highlight the locations of some of the nuclear pore complexes. This is a portal between the inside of the nucleus and the outside. Proteins and RNA are going back and forth. It's about 120 nanometers in diameter. It consists of somewhere between 450 and 500 distinct polypeptide chains. There's no way this is ever going to crystallize. But by combining X-ray structures that we do have, such as the structure we determined of NUP190. They're all called NUPs, the nuclear pore complex proteins. Combining structures such as the structure of NUP192 that was over 1,000 amino acids determined as part of our NIH-funded structural genomics effort, with the results of electron microscopy, small angle X-ray scattering, cross-linking, sedimentation, affinity purification, methods like FRET, etc., the use of homology models, and then combining all of that with the use of a technology called satisfaction and spatial restraints, it's going to be possible for us to produce atomic structure models of large assemblies such as the nuclear pore complex, admittedly with a lower precision than X-ray structures. So I told you early on that a typical protein structure like that one, the precision of the XYZs is about 0.2 angstroms, 1/10 the resolution limit. The precisions of the models that are going to come from this hybrid approach are lower, to be sure, somewhere probably between 3 and 5, 6 angstrom in terms of the uncertainty of an individual atomic position. But nonetheless, a source of hypotheses with which to do studies to understand how this machine works as a regulated gate that allows entry and access of proteins and nucleic acids to the nucleus and from the nucleus into the cytoplasm in a way that ensures proper regulation. The other thing that I think is going to happen in the next 6, 7 years is that it might be possible for crystallographers of macromolecules to put themselves out of business. And by that I mean that with the emergence of the free electron laser and doing diffraction from a single macromolecular complex in solution, we might be able to stop having to crystallize anything. So this is a pipe dream at the moment. There's a free electron laser operational at Stanford today that has a repetition rate of 120 hertz. There's a flash 120 times a second. And there's a team that has developed a liquid jet delivery system for nanocrystals. So this is like an inkjet printer, but it's shooting a column of liquid that contains protein crystals into the beam, and every now and then the crystal's in the right place when the X-ray beam flashes. You record a diffraction pattern, you pool all this data for many, many hours and many, many milligrams, hundreds of milligrams of protein crystals rushing past, each one of these crystals very small. You can recover the 3D structure. In 2015, we believe in 2015, the free electron laser that's being jointly funded by Russia and the European Union, that's why it may not come in 2015, is going to come online at Hamburg. And that's going to have a repetition rate of many thousand hertz. So that's going to greatly increase the collision rate between the X-ray beam and the nanocrystal and allow for much more rapid measurement of X-ray diffraction patterns from crystals, very, very small crystals of things that might be very interesting for which one can't grow a big crystal. Now, I happen to believe that growing a small crystal is almost as hard as growing a big crystal, because nucleation is the gating event in crystallization of macromolecules, I would argue. And so as a result, I think that this approach is going to have limited utility. But if you have a very, very, very short, very, very, very intense pulse coming from a free electron laser, a femtosecond pulse, you can record the diffraction pattern of a single protein or a single macromolecular complex in solution before the protein burns up. The diffraction event occurs, is recorded by the detector. The protein is then denatured, of course. Denatures very, very soon after that femtosecond pulse is delivered. But you've recorded the continuous Fourier transform of the protein in that particular orientation. Now, this is an X-ray laser. That means that the X-rays are coherent. That means you can do phase recovery. That means you can overcome the phase problem. So now how do you deliver the sample? How do you pop a single protein up into, or a single complex up into the beam when the X-ray beam arrives, when the flash arrives? Well, the answer to that question may lie in the hands of experts like Peter Lobel and his team downstairs. Ion trap mass spectrometry could potentially be used to trap ions next to the X-ray beam using a particular M over Z, and then to eject one ion at a time out of the ion trap mass spectrometer into the X-ray beam. record the diffraction pattern of the complex in all possible orientations, invert all those transforms, and come up with an electron density distribution for the protein. Mathematically, this could work. Whether or not the laser is going to be bright enough, whether or not the pulse is going to be short enough, we still don't know. But I'm betting on the fact that this is going to revolutionize structural biology in 2020 and beyond, because we will be able to study all sorts of things. And this is going to give cryo-electron microscopy a run for its money. So let me close by saying that these advances are going to create challenges for the protein data bank, but challenges that we want to confront. So we are going to foster development of a network of macromolecular experimental data resources that include protein X-ray crystallography, of course, because we want to enable PDB deposition of structural models derived from integrative methods so that we can capture assemblies of the size of the nuclear pore complex and other really exciting machines that operate in nature. So that's our commitment, collective commitment to you as a community of structural biologists. And I just want to close by acknowledging the many influences that I was the beneficiary of over the, 35 years I've been doing structural biology. They're listed alphabetically. The underlined names are those of women. The names with an asterisk are those who won the Nobel Prize. And anybody who's there in green was a member of the Howard Hughes Medical Institute. There's a lot of them. We all stand on the shoulders of giants when we do science. So I apologize for going a little bit over. Thanks very much.
Thank you, Stephen. That was a wonderful talk. Have a little bit of time. Stephen.
Yes, sir. The brave one. Well, you have to decide you don't want to sleep. That's the first order of business, is doing without sleep. Now, one thing you learn when you're an intern and a resident, you can do without sleep, but you can't do without food, which is why they feed you at 10 o'clock at night to keep you awake all night, taking care of the patients when you're on call. I've been very lucky. I've had a series of fortuitous opportunities that I've capitalized on, and I think the key thing, what advice would I have for young people starting out today? Well, there's the Monty Python advice, which is you get up 3 hours before you went to bed, you lick the lab clean, and then you do world groundbreaking experiments. So that's the Monty Python view of how to get ahead, but that's not real, of course. That's you obviously don't know that skit, do you? In fact, most people don't seem to know that skit. These guys sitting around smoking cigars, obviously they made it. And they say, oh, it was so tough when we were young. I remember we used to have to get up at crack of dawn, and it would be raining. And then the next person, oh, I had it much worse. I had to get up 3 hours before I went to bed, lick the street clean, go to school without any clothes. I mean, it just goes on and on like this. And the culmination, the punchline, of course, is the problem with young people today is they wouldn't believe us. So leaving that aside, I think the key is you want to choose really interesting, really challenging problems to work on. There's no point in working on something that's me too. And you have to make a total commitment. And that total commitment's really scary, because if you fail, wow, I tried. I maxed out and I didn't get there. But that's a risk it's a risk you have to be willing to take, I would argue, to do great things in any line of work in life, and to achieve. But everyone who's here has probably been an A student through most of their school career. They're used to working hard, they're used to achieving. And so then the other piece of advice, in addition to hard work, I would give is, this comes from Pasteur, so paraphrase is, nature favors the prepared mind. So when you see the data, be open to what the data might be telling you. Don't go in there with a preconceived chance. Thank you. Chance favors the prepared mind. Thank you. you might see something interesting that's really, that changes, the way people think about science, the way people think about nature, business, finance, whatever. Yeah.
I'd like to thank Stephen again for a very, very